won't be on camera, but we will be recording it and putting it onto the ERA4TB video channel, and that will happen within a couple of weeks after the session, once we've reviewed and got the transcript together, and then that will be communicated via our ERA4TB social media channels, which you can see on the side here. We have an X or Twitter channel and a LinkedIn channel. So if you are following along and want to post anything, do tag us in on those channels. Only our presenters' cameras and microphones will be available today, so you won't be able to speak directly, but you can ask any questions. There's a Q&A function, so that will be a button either at the top or the bottom of your screen, depending on the device that you're using. And we really encourage you to send any questions you have for our speakers as you hear the presentations, and we'll answer as many of those as we can at the end of the session. So very, very quickly, just if you haven't heard of ERA4TB before, we're a public-private initiative funded by the Innovative Medicines Initiative. The main objective of the project is to create a European open platform to accelerate the development of new treatment regimens for tuberculosis. We're running for 6 years, and we have a progression pipeline that caters for a variety of molecules at different stages of drug development. As I said, we've got 3 speakers today. So kicking us off will be Juan Jose Vaquero from UC3M, who'll be talking about preclinical molecular imaging protocols. Following on from that will be Silvia Grandoni from the CNR in Rome, and she'll be looking at model-based approaches to anti-tubercular drug development. And finally, we'll be hearing from Patrick O'Meara from the Critical Path Institute, talking about the work that's been done to develop a drug development information management system. So without further ado, I will hand over to Juanjo to start the presentations.
Thank you very much, Kat. I cannot turn on my video. I guess you are controlling my video. So may I have the first slide, please?
Of course.
Biomarkers derived from animal models are one of the keys to define functions for predicting the efficacy of new treatments in humans. So next slide, please. The introduction of biomarkers based on translational medical imaging modalities increases the power of the predictive models while improving the experimental condition of the animals, because its subject is his own control in monitoring the effects of the treatment until the end of the experiment. Next slide, please. So my name is Juan Jose Vaquero. Next slide. I work at the Universidad Carlos III de Madrid, and in this talk I'm going to talk about, I will describe the preclinical and molecular imaging protocols for TB regimes that we are developing in ERA4TB, and focus on the translational imaging-based biomarkers. Next slide, please. Well, let me start with a very simple example in which you have on the screen 2 images of 2 different animals. On the left-hand side, you have a CT render in 3D render of the space occupied by the lung of the animal. And you see you can recognize the shape of the lungs, the ribs, etc. And basically, there is nothing else. It's an empty space. However, if you look at the right-hand side picture, you will see that inside the cavity in this animal, there is a cloud of red and yellow. That's the render, the 3D render of the PET imaging in this animal. The PET imaging has been done with a tracer. In this case, is the fluorodeoxyglucose, and what it's doing is labeling or is highlighting high rates of metabolisms in the space in the lung. And in this case, this increase of metabolism is related to an infection process. While in the left-hand side picture, you don't see anything. There is also a 3D render there of the PET, but there is nothing because there is no a condition like similar to the one we have on the right-hand side. So next slide, please. So then you see that the value of the imaging, just looking at that, there is a lot of information that we have there. So if we can quantify what we are seeing, we will have a lot of information, and the validity and the reliability of the data that we'll extract is that's the real value of doing imaging, molecular imaging in our models. Next slide, please. So now, based on those principles, and I will focus just at the beginning just on CT, we can monitor the animals. That's one of the advantages of imaging, is that the animal, we can repeat these sessions and take the same measurement along the progression of the disease. So here you have 3 pictures of the same animal, first at the beginning, before the animal was infected, and then 8 weeks post-infections, and then 12 weeks post-infection. And you can see very clearly several things. This is a sagittal slice over the thorax. You can see the lung delineated in the before injection image. When we moved to the 8-week post-injection, you have seen that the lung have growth, okay? And also there have been changes inside the lung itself. And when we go to the last week, some other changes have happened. And we can measure this evolution of the disease or over the effect of the treatment over time. We can quantify on each of the time point and get all those valuable data. Next slide, please. So now, based on this power that of imaging across time, we can define studies and protocols like this one. In ERA4TB, we work with 3 animal models. One is the mice, and the other one are 2 non-human primates like the marmosets and the macaques. So this type of studies is similar for the 3 of them. In the upper panel, you see the timeline from the beginning before the animal is affected to the end of the experiment, and the red, blue, and green are symbols that mark what type of data acquisition are going to do in the different time points. So you see that we draw blood from the animals. We measure bioclinical science and other parameters along the experiment, and we also take a certain point PET-CT images. And so we measure bacterial load at different points, and as you say, at the end, at the very last time, we can get samples of the different tissues of the animal and do histology. That is really, in histology, we are measuring parameters with great relevance for the clinical evaluation of the animals. Next slide, please. So for each time point, we will need to extract these biomarkers. In this case, there are different approaches. This first approach is to analyze the whole lung, the lung as a whole. And then inside that lung, you see that over time, the composition of the tissue changes. What we do is we do an analysis, an automatic analysis that accounts for that change of tissue composition, and we can separate in 3 different types: the healthy tissue and the diseased tissue. Inside the diseased tissue, there will be 2 components, what we call the soft and the hard tissue. And by using this automatical method, you can see that we can, at the top above, you see the CTs, under that you see the colored coding for the different type of tissues. Once we get that, next slide. When we get that, we can quantify the volumes. And if you look at the left-hand side panel, we can correlate with measurements that are the ones that are currently being used to evaluate the progression, like, for example, the CFU. And you see 3 different groups. You see the control, excuse me, one second. You see the control animals that with different size in blue. You can see an infection control that is not treated, and then you see that has a very high load of CSF with different sizes or different volumes of the uninvolved tissue, I mean the healthy tissue. And then you see an animal that has a series of animals that has been treated, how they maintain the same level of infection by the healthy tissue recovers over time. it probably is easier to look at that in the right-hand side panel, in which you see the evolution of an uninfected mouse over time, in which the relative healthy tissue, because it's not sick, nothing is changing. While when you see on the darker blue plot, in which the animal has been infected, you see as time progresses, the volume of health tissue is reduced. And that means that the disease is progressing and is taking more, colonizing more and more healthy tissue. That could be just because it's not treated, or just because the treatment is not effective. So it's a very clear quantification that we can use to follow up quantitatively what is going on. Next slide, please. So, although these metrics looks very nice, we have some problems, and even though with automatic methods and artificial intelligence methods, we have to face the inconsistency of these mixtures. As the disease progresses, there is a higher mix of different tissues that make difficult this different segmentations. And also, we have to cope with artifacts that we have, like, for example, the breathing. The animal are alive, so they keep breathing, and the breathing movements move the lungs and blurs the contours of the different tissues, and it's very different to segment. So as the infection increases, we have more difficulties in getting the same quality measurement over time. So in order to solve for this, next slide, we have been aiming for other type of measurement, like to identify individual lesions in the lungs, count them, follow them over time. As you see here, 4 panels, 4 time points for the same animal, in which we have identified in blue a specific manifestation of the disease that we follow on time, and we can measure how they grow, how they reduce, and how new manifestations appear. Next slide, please. These manifestations are segmented by expert radiologists, and expert radiologists that can do this type of segmentations or analysis in macaques, marmosets, and mice are not many of them, so it's difficult. So if we want to automatize this type of analysis, we will need to have enough data sets as a ground truth that will allow us to train our models to be able to do the same segmentation. So that's the first problem. We don't have a lot of data sets in which we can rely for that. And second is that even if we have 2 very well-trained doctors to doing these segmentations, they may have differences. Because you see on the left-hand side, user or radiologist number 1 has identified a mass as one type of manifestation that radiologist number 2 is not considering of the same class. So that means that still there is a lot of work in order to have a ground truth in which we can rely on to develop our models. So what we can do if with these difficulties, what can do to improve our biomarkers is to add PET. Next slide, please. PET is a modality in which instead of analyzing the morphology and the density of the tissues, what is doing is to display the activity of the certain tissues and certain cells according to the tracer that you have injected. There is a lot of literature saying that different tracers could help to do PET-CT, PET imaging in these models. Next slide, please. So we have adopted using FDG, that is the most widely available tracer, and is the same one that I showed you at the beginning, is the same animal, and this is the result, a PET-CT, in which we will combine both analyses, the CT that I just explained, and a very similar analysis of detecting these conglomerations, these clouds of activity inside the lungs. You see the PET here in red and yellow also, and you see the different areas, how the uptake changes depending of the condition of the tissue, each of them. So the same type of segmentation that we showed you before for the individual manifestations applied to PET are the techniques that we are using. Next slide. In the case of PET, we have to deal with the non-specificity of FDG. FDG, as I said, is a metabolism marker. So it will highlight those zones with higher metabolism, like you can see very clearly in the left-hand side, an animal. You see the brain highlighted, you see the muscle, you see the heart, that is that brilliant ball in the center. And this non-specific uptake can confound or make a noise that limits our capability for segmented, to separate the activity due to the infection in our case, in the case of TB. So we have to deal with this, and there are several ways with some drugs that we can inject, or even just changing the diet of the animals. We can switch the metabolism from fatty acids from sugar to fatty acid. And then by doing these protocols, you can see in the right-hand side, an animal with a diet type B, in which the non-specific uptake has been mostly reduced. And now, if that's a healthy animal, that's what it shows, nothing in the lab. If that case were an infected animal, we will highlight those manifestations of in the PET in the same way that we saw on the first slide I showed you of the guinea pigs. Next slide. So another way to solve this problem of the specificity in PET is to say, okay, then instead of using this generic FDG, can we have any specific tracer that will help us to measure more quantitatively, more precisely, where is the disease? Well, there is a tracer that is called the deoxyfluoride-3-allose that is very specific, has a very specific Mycobacterium, has a very specific uptake for that. That's a great tracer, because that will allow us not to see the infection, just to see where the alive bacteria are in the lungs of the animals. So this is a project that we are starting in ERA4TB to try to increase the specificity of these biomarkers, and this is an action that we are trying to add to all the pipelines that we have shown you that we are doing today. Next slide, please. So just trying to summarize, what we are doing in ERA4TB is to compiling and building tools very specific for the different models, because there are different conditions, different size, and even the anatomy change in between among the mice and the two non-human primates. We have built these different type of pipelines. Our image producers can upload the data sets, and we can run these pipelines and produce automatically those biomarkers based on imaging. Next slide, please. And just to conclude, if I have to wrap what we are doing is in 2 sentences. In ERA4TB, we have established a series of workflows or pipelines to derive or to extract image biomarkers as a tool to study new treatments for TB, thinking always in the translational effect. What we are doing in the animals is very translational at what we doing in humans. We are treating the animals exactly the same way that we do to humans. So those no invasive biomarkers that allow us multiple readouts has that powerful or be translational. And our goal, as I say, is to develop these biomarkers, adding more and more specificity of them to become predictors of treatment, of efficacy of treatment in humans. That's our final goal that we will to achieve by the end of the project. Before ending, next slide, please. I want to thank to all the people that is contributing to that. Here I'm listing the drivers, but there is a lot of more people behind us in not only in these institutions, in other groups that are in ERA4TB, and thank all of them, and thank you for your attention. Thank you.
Thanks, Juanjo. That's great to see. And again, just for anyone who's joined, there is the Q&A function if you want to put any questions in for our speakers today, and we'll try and get to those at the end. So moving on, we will now go on to Sylvia, who's going to be talking about the use of model-based approaches for drug development.
Yeah, thank you, Catherine, for the introduction. So I'm Silvia Grandoni. I'm part of the modeling and simulation team of ERA4TB. And today I would like to talk about how modeling and simulation can be used for a more rational design of drug combination and drug development. Next slide. So the treatment of drug-susceptible pulmonary TB is still quite long, so it requires 6 months of treatment involving 4 drugs. And these cause problems in terms of adherence, high cost, logistic burden, and acquired resistance. Next slide. So there have been really incredible progresses in terms of development of treatment for TB, because now we have a 4-month regimen that has been proven to be non-inferior to the standard of care. And we have also a very rich pipeline with a lot of drugs that can be potentially combined to provide shorter and more efficacious treatment for TB. Next slide. So the treatment of TB is very challenging. And one of the reasons is that MTB is able to survive to the stress induced by the immune system. The immune system, to try to contain the infection, causes the development of several lesions, and these lesions represent a niche for the bacteria. The bacteria change the metabolism to try to resist, and the result is that we have MTB present as multiple phenotypes with different drug susceptibility. Next slide. Another important aspect that has been neglected in drug development for TB is the penetration in lesions. So lesions are niches for the bacteria, and the drug needs to penetrate inside the lesions to kill the bacteria. So it's important to take into account the drug exposure into lesion to be sure that we are providing potentially sterilizing treatments. Next slide. Another important aspect that we should consider is the immune system response as an active player in controlling the infection or in clearing eventually the residual bacteria remaining after the treatment, preventing relapse. So taking into account this element can contribute to the treatment shortening. And in addition, we know that the co-infection MTB HIV is an important health problem. And the co-infection with MTB is the leading cause of death in individuals with HIV. So it's important that we are able to take into account the immune system response to try to also provide effective treatments with people with weakened immune system, like in case of HIV. Next slide. So having said that, it's clear that shorter sterilizing treatments will come from drugs with complementary properties in terms of penetration into the lesions and activity against the subpopulation. And given the fact that there's no one single experimental model that is able to recapitulate all the physiological features of MTB in humans, the development of the treatments cannot rely in single experimental models. So we need to integrate the data coming from different models. Next slide, please. So with modeling and simulation, we have the opportunity to do this. So within ERA4TB, we have a lot of preclinical models. for example, in vitro experiment, we're using in vitro experiments in which bacteria are exposed to constant concentrations as high throughput screening or low throughput screening. Experiments in which the bacteria are exposed to concentration varying over time. And we have different models, animal models, that are helping us in understanding specific aspects of the pharmacokinetics and pharmacodynamics of the different drugs. So with modeling and simulation, we can help scientists in clarifying the pharmacokinetic pharmacodynamic relationship of drugs and combinations in the different stages of the drug development. But also we have the opportunity to provide robust translation of the results across the different phases of drug development, trying to predict as soon as possible the outcome of the treatment in humans. Next slide. So traditionally, the assessment of the pharmacokinetic and pharmacodynamic relationship was based on the link between the bacterial burden assessed in the lung, usually through CFU count, and the systemic drug exposure, so the exposure that is assessed through blood samples. But this is too simple to characterize, to take into account all the complexity of TB and to ensure a robust evaluation of the pharmacokinetic and pharmacodynamic relationship. So for that reason, we need to integrate all the data coming from different experimental models related to the pathogen, to the drug, to the immune system, to help the drug development process. Next slide, please. So I would like to start showing how with modeling we can put together the pieces of this puzzle. And I would like to start with showing examples of models related to the drug and a pathogen in interaction. Next slide. So several models have been developed that are able to describe the bacterial dynamics, including the different phenotypes, as I showed before. We need to target the different phenotypes to be sure that we achieve sterilization. So these models are the basis for the assessment of the drug efficacy and quantification of drug efficacy against the different phenotypes. Next slide, please. So this model can be applied within different experimental settings. So this is an example in which a multi-state bacterial model was applied to analyze time kill assay data, so data in which bacteria are exposed. So in time kill assay is an experiment in which the bacteria are exposed to constant level of concentrations. So with this model, it was possible to characterize the efficacy of the drug against the different phenotypes, but also the pharmacodynamic interaction. And this can be used as a basis, for example, to prioritize combinations at a very early stages. Next slide, please. Another approach, still including the dynamics of the bacteria, of the different phenotypes, has been applied, for example, to in vivo data from the mouse model to quantify the effect of the drugs against the different phenotypes and to understand the impact of the drugs in the different combinations in the combination. So this was used, for example, for the purpose of combination ranking. So this model has the impact has an impact in the way we can assess the combination in the preclinical stage for the purpose of ranking, but also for subsequent extrapolation. Next slide. So another aspect that has been neglected but can be better characterized with modeling is the exposure at the site of infection. Next slide. So next slide. We have different modeling approaches that we can apply to characterize the pharmacokinetics in the lungs in preclinical species. And then we can translate these into humans to try to predict the exposure, the lung exposure in humans. So this is an example in which, for example, PBPK modeling was used. So with this approach, the translation is easy because we can update the physiological parameters of the model according to the species of interest. And we can keep the parameters related to the drug fixed. So we can develop a model with the data available of lung exposure in a preclinical species, and then we can update the physiology to humans to try to predict the exposure in lung in humans. Next slide, please. So within ERA4TB, we are working hard also with not only with the modeling team, but also with other work packages to expand this approach to be able to describe the drug exposure in lesions, because as I said, is where the most resistant bacteria reside. So it's important to take into account this for also for further translation of the PK/PD relationship in humans. Next slide. So I'd like now to focus on immune system. Next slide, please. So as I said, it's important to take into account immune system response in anti-TB drug development. We have models that are able to describe the immune system response in humans. As I said, there's no one single model able to characterize the immune system response in a way that is really close to that in humans. So we have this platform that have been developed for many years, in which the interaction between a pathogen and the different players of the immune system can be modeled. So now we are working within ERA4TB to try to customize this model that was originally developed to describe the immune system response against some pathogens, to take into account the specific immune response against TB, including also the different phenotypes that are present. And with this modeling approach, the different players, the interaction between different players is coded through different rules. So these rules can be updated on the basis of the interaction that we want to describe and simulate. Next slide, please. So these are preliminary results. So the model has been calibrated to be able to reproduce epidemiological outcomes. In these results, we see that if we simulate a cohort of patients and the administration of the standard of care therapy, the bacterial clearance in terms of time to achieve the clearance of the pathogen is consistent with what is observed in clinical trials, but also is consistent with other in silico simulations. So this is a very positive result. Next slide, please. So with the same approach, so still using an agent-based modeling approach, we're also working to describe the in silico, the formation of granuloma in vitro. So we have been working to code the interaction between MTB, monocytes, T cells, cytokines. And the model is able to reproduce, to simulate the formation of the granuloma in vitro. And this has an important implication because we can help the experimentalists working with in vitro systems to better understand the PK/PD relationships of the drug, take into account the size of different granulomas, which is not possible in vitro for experimental limitations. And this will be the basis also for further description in silico of the formation of the granuloma in vivo. Next slide, please. So to conclude, I would like to show some translational application of modeling and simulations to evaluate the treatment performance in humans. Next slide, please. So as I said, these models have the potential to be applied across different experimental settings to try to predict the outcome of the treatments in humans. So, and integrating different data that are generated along the drug development process. So in this example, the model taking into account the dynamics of the different bacterial phenotypes has been applied to in vitro data to characterize, first of all, the potency of the drug against the different phenotypes and to characterize the pharmacodynamic interactions, and then integrating the human pharmacokinetics and some translational factors, the model was able to successfully predict the early bactericidal activity in humans. Next slide. In another, we can see here another example in which a similar approach, still using the bacterial dynamics modeling, has been applied to mouse data to characterize the PK/PD relationship of drug combination based on bedaquiline. So after the characterization of the PK/PD in the mouse using integrating the human pharmacokinetics and some translational factors, it was possible to correctly predict the early bactericidal activity in humans. So these approaches have the potential to change the way we design the combinations to predict as soon as possible, and we can predict as soon as possible the outcome of the treatment in humans. Next slide, please. So I would like to conclude with some take-home messages. So in the past, the development of new combination regimens for TB has been mainly based on an add-on and replace strategy based on the standard of care. However, we have now new methodologies, so new experimental models and also new in silico approaches to have a more rational evaluation of the PK/PD relationship of new drugs, but also of combinations. And it's essential to integrate all the data coming from the different experimental models to ensure robust translation of the findings across the different stages of drug development. And with modeling and simulation, we can integrate the data and we can try to predict the efficacy of the combinations in the clinic from the early stages, reducing uncertainty before implementing clinical trials. Next slide. So I would like to say that all these progresses we are doing within ERA4TB are the result of several interactions. So within the modeling team, so the CNR team, and I'm part of this team, but also with the Uppsala University team, which is part of the modeling and simulation group of ERA4TB, but also with the work packages and with the asset owners. Next slide. So I would like to say thank you to everyone for the very fruitful discussions we had along this journey within ERA4TB, and thank you to you for listening.
Thank you so much, Sylvia. That was great to hear, and I think that takes us nicely into Patrick's talk. We'll be looking more at data integration. As we've seen, it's a very important factor in the project. So, Patrick, I will hand over to you.
Okay, thank you, Catherine. So just as a brief introduction, my name is Patrick O'Mara. I'm a data team manager from the Critical Path Institute, and I work within the data and pipeline management work package for ERA4TB. And on behalf of the ERA4TB team, I wish to give a fairly brief overview regarding the Drug Development Information Management System that has been developed and utilized on this particular project. And during these, say, 12 to 15 minutes, I'll be touching upon the purpose and objective of the DDM itself, the considerations and factors leading to its setup and ongoing development, and also brief a touch upon the various features or platforms that are residing within the DDM system itself. Next slide, please. So in terms of the purpose or objectives of the DDM, so the intention of the DDM is to incorporate complementary technologies into a centrally accessed control system, which in turn is to be established to support the progression of the various pipeline development activities within the ERA4TB project itself, but also to ultimately to have the data generated from these efforts to be suitably standardized and where possible linked together within the system to ensure enhanced accessibility. Next slide, please. So in terms of the progression pipeline, so the progression pipeline itself caters for the development of molecules at different stages of development within the project itself. And as a result of these molecules or even combination therapies being at different stages of development, the DDM itself is tasked to be able to incorporate various data streams and data types from various institutions involved in these pipeline activities into the system itself. So just to very briefly mention what we're receiving in, we're expected within the DDM to receive clinical trial data, in vitro and in vivo data experiments, CMC, GLP and non-GLP data, as well as preclinical images, specifically the tabulated data associated with those images that are going to be processed during the course of this project. Now, due to the variety of the data being handled by these various partners, groups, and institutions, this does not simply just involve acquiring the information from these partners as reported by the institutions and just inserting it into the DDM as is. In addition to that, it's also necessary to have the processes and tools available to ensure that both the data and associated supportive documentation within the DDM are consistently structured in terms of tabulation, terminologies, and values used, that the appropriate key identifiers have been implemented to ensure that, and to aid, may I add, that you can link and correlate various pipeline activities that are happening concurrently together, and ultimately to optimize both the interoperability and reusability of that data, which is available within the system itself. Next slide, please. So with these data stream considerations in mind, the initial setup and the ongoing progress and development of the DDM itself was reliant on various interdependencies and rate limiters to move the system forward. So these factors and challenges include, from a reporting perspective, identifying what data is being generated, how is such data being reported or expressed by the various institutions, and as a consequence of that information, determining the best means in terms of getting templates developed or worksheets developed to have the information better structured and made available for processing into the DDM system itself. From a data consumer perspective, ultimately is to understand what the end user or the consumer wishes to see in the systems themselves, and also receiving specific requirements on how they wish to interact or utilize the data or the systems themselves. And then on top of that, due to the fact that a number of these molecules are still in early phase of development, and also we're dealing with human trial data in some parts of the pipeline activities, there needs to be a clearly established governance process which takes into consideration topics such as data privacy of the subject in terms of clinical trials, but also IP or intellectual property considerations in terms of how and whom can access these specific data sets, and how that data set is going to be utilized further downstream. Now, last but not least, from a technological or technical perspective, the technical groups essentially have to understand and figure out essentially how to integrate the various technologies into this single system, i.e. the DDM. But in addition to that, the feedback received from the data reporters, the consumers, and access governments also has a bearing or influence of how the system is set up. So it's a combination of these 4 factors, which first of all needed clarification before we actually were in a position to actually initially set up the system and also to further progress the system as the project has moved on. Next slide, please. So based upon these interactions in the previous slide, the current net result is that the DDM has progressed to include 5 distinct components, which are namely a central single sign-on portal, which enables end users or specifically authorized end users to access the various tools that are made available within the DDM system itself. These include the Grid42 data platform, 2 imaging platforms to support different types of formats of images, which are being generated as part of the pipeline activities, as well as a management or oversight dashboard to provide oversight of the various pipeline activities that are being conducted within the project itself. And in these next subsequent slides, I'm going to go into a little bit more detail about some of these components, just so you get a sense or flavor in terms of what those systems are doing. So if I could have the next slide, please. So in terms of data tabulation, all results from the pipeline are to be standardized and integrated into the Grid42 system. And to date, for everyone's information, we have 38 distinct pipeline activities which have been integrated. And with all these data sources mentioned, the end user has the ability to be able to view and browse the data within the actual Grid42 platform, able to filter it, and in addition to that, are able, based upon those filters, subsequently export that data for further downstream use, i.e. analysis. So there is some filters within the platform itself, which enable, say, a modeler or an analyst to kind of cherry pick, if you want to call it that, the various data that they need to support certain activities. In addition to those, say, fairly rudimentary capabilities, there is a capability of actually creating a structured activity relationship tables or SAR tables that allow the ability for end users to compare compounds of a specific selection against specific assay endpoints. And more recently, we have released, with obviously the efforts of Grid42, a new version of Grid42 into the DDM where there's an application programming interface, which in layman's terms facilitates authorized users to be able to connect to Grid42 using another app. So for example, as potentially a statistician or a modeler, you could actually utilize a tool like, say, RStudio to connect directly to the Grid42 system without actually being in Grid42 and be able to analyze or interrogate the data for whatever specific objective and purpose. Next slide, please. So briefly speaking upon, I guess, imaging itself, and Juanjo went into detail on some of those topics. From the perspective of PET-CT and MRI images, which are of a DICOM format, UC3M have decided or came to the conclusion to utilize the XNAT imaging platform. This is an extensible open source imaging informatics platform, which also possesses advanced data management and analytics capabilities. And based upon those features, most importantly, that because of these, say, advanced DM capabilities, for instance, it supports the ability to actually organize the information or data into tabulated outputs, which in turn can support ease of integration into Grid42, as well as creating, say, various reference links, which can be utilized for navigating back and forth between the 2 platforms. So again, improving, say, the capabilities of accessibility. To date, just for everyone's awareness, we have received some CT scans. However, we're still under discussions with the relevant partners in terms of finalizing the data tabulation piece. So this is currently a work in progress. Next slide, please. So in addition to XNAT, there is also a need to be able to handle and integrate, for want of a better word, non-DICOM images. So within the ERA4TB project, we have time-lapse macroscopy, MALDI, ion beam histology, which were planned to be conducted during part of the pipeline activities. So in light of those needs, UC3M have opted to create a separate repository which structures the images in a specific hierarchy, which is based upon per pipeline activity, then on a level of experiment ID, and then on a subject level. And it is our intention, and may I add, this is still a work in progress, is to use the hierarchical structure within the actual imaging repository to actually link to the tabulated data that will eventually reside in the Grid42 platform. So, I mean, this is still a work in progress, and we would imagine that, say, within the next, say, 12 to 18 months, there'll be further improvements to regarding this particular process. Next slide, please. So finally, in terms of the components, I'm just going to focus on for today, there is the pipeline dashboard. And so just for everyone's awareness is that on the ERA4TB project itself, we do have a large volume of pipeline activities, both ongoing and in the future that will start. Currently, I believe there's somewhere in the region of in excess of 100 distinct activities, which are either ongoing or planned. And so as a consequence of that, it is important that a specific tool is being developed to support from a, I guess, from a project management perspective, a tool to allow the oversight of all these different moving pieces. So UC3M again have actually supported in the development and ongoing development of a dashboard, which will support project management groups in terms of overseeing the activities in the pipeline. So this includes being able to filter information on a work package perspective, partner perspective, on a molecule level, and also be able to filter on a data level to kind of identify what are the activities within a certain window of time. And again, this ultimately has been developed to support oversight tracking. And this is obviously quite important to bearing in mind that the volume of pipeline activities that are actually currently ongoing and expected to continue in the future of this project. Next slide, please. So just to summarize and conclude, the current efforts to date in terms of what we have achieved with the development and the setup of the DDM and its ongoing enhancement is that it's basically our intent to try and develop a one-stop shop to aid in the collection and organization of data to support decision-making and or analysis of the pipeline. It's my view, at least, that this approach has aided in, one, fostering increased harmonization of how data is presented to support improved interpretation. Via such harmonizations, it also enables a means to link concurrent activities across various tasks and institutions. So, i.e., if there's parallel activities done by institutions which are complementary, these can be linked for analysis purposes quite easily based upon the key identifiers that have been established within, say, the data and the systems themselves. And again, because of these structures and frameworks and the templates and worksheets that have been developed as part of these efforts, it also encourages and informs researchers to follow a more consistent approach in reporting as well. And finally, with that consistency and with the oversight tools available, it also, we feel, can support the progress and oversight of the various regimens and compounds at various stages of development. So just as a note that what we've presented here is still a work in progress. There is a lot of data that's already been received. However, with the number of pipeline activities still ongoing, there is the possibility that further enhancements are expected to occur with these various components. I think more specifically, and for example, from an imaging perspective, we are only starting to actually receive data in or images in for the purpose of the various pipeline activities. So from an imaging perspective, especially, I would envisage there will be further enhancements, improvement, and progress in the future regarding the actual DDM system to support both ERA4TB and potentially other activities in the future. And I guess with that, that concludes my presentation of DDM. And I guess we're now moving to the QA section. So thank you for your time and attention. Thank you.
Thank you so much, Patrick. Thanks to all the presenters. I think we've heard a really interesting selection of activities that are going on from kind of the really interesting, innovative imaging data that's being collected, how that's then being integrated into a management system, and then obviously how people like Sylvia are taking these different data sources to using computer modeling to do prediction based activities. So just having a look at the question to the presenters. So Patrick, if I could perhaps just start with you. I was just wondering, this is really fantastic to see all this information being pulled together. You know, are there opportunities for external partners to be able to access that information in any way, or are there any other sorts of, I guess, initiatives going on to allow that to happen?
Presently, no, but that's contingent of an external group coming to ERA4TB with a compelling objective which aligns with what we're doing essentially. So to give an example, and I'm not sure if there's members of this particular consortia here, but for instance, Unite4TB, which essentially is from a pipeline perspective, is essentially the natural successor to ERA4TB. There is potential power for members of Unite4TB to gain access to some of this data to perhaps support some of their goals and objectives. And, you know, as you probably noticed or are aware of, that there are some assets from Unite4TB that actually have been put into the pipeline here as well. So there is, I think, a possibility for, say, Unite4TB to be, you know, or at least aspects of Unite4TB to be kind of, you know, crossover into the DDM. However, whether it happens now is contingent to the relevant partners to contact us. And when I mean us, I'm not talking about, say, myself personally, but actually the PMO itself, to figure out what is needed and also come to a consensus of how this would work in terms of collaboration between those projects. So in theory, yes, but the contingent to that is, you know, what is the use case and how is that going to be worked from a logistic and procedural perspective?
Great, thanks, Patrick. So if anyone is interested in that, then obviously please go to our website. You can find our contact details there, and be happy to kind of start those conversations. Juan, perhaps I can go to you. So really fantastic to see all this work that's been going on with imaging. Like how are you kind of using the imaging biomarkers with other types of biomarkers? Like what's going on? Is there work going on there to kind of look at all these different biomarkers together to kind of predict outcomes for patients, or sorry, not outcomes for patients, but drug effectiveness?
Well, first thing is we are working in extracting these biomarkers and trying to compare with the traditional biomarkers. So the first step we have to achieve is the approval of the new biomarkers. Okay, and then the value of the image-based biomarkers will be to integrate it in the models that Sylvia has described.
Mm-hmm.
So I don't think that we will have a direct application from the image biomarkers directly to the conclusion of the effectiveness of the treatment. I think those are data that will be added to the current collection of data that modelers use just to do this prediction. Of course, you always dream into finding the holy grail and get this signature that will tell you immediately if this is going to happen equally in humans. But being realistic, not a single biomarker based on imaging will be able to do that, at least by now. We will see what other things we can develop over time, and what are the results of the new approaches based on artificial intelligence. So we will see.
In the future. Fantastic. Thanks, Juanjo. And lastly, Sylvia, just before we run out of time, I just wanted to ask you kind of what you think could perhaps support the development and application of these modelling frameworks.
So yeah, I think that first of all, the communication between the different teams is very important. So we need to be able to explain what could be the value of what we are doing to the people doing the experiments, because sometimes we require more data, more effort. So we need to be able to communicate the potential value of what we are doing. So, and also the development of new biomarkers, or the implementation of some cutting-edge technologies and biomarkers, for example, to assess the presence of phenotypes in vitro and in vivo experiments, or the images to understand the penetration of drug into the lesions are things that can be combined with within our modeling frameworks to give the prediction of what will be the treatment outcome in the clinic.
Great, thank you. It sounds like it's a really exciting area moving forward, and with more, with all the activities going on in ERA4TB, the new data, it will just become more and more interesting to work in that area. So thank you. So we're at 2 o'clock, so it just leaves me to say a big thank you to our speakers today, Juanjo, Sylvia, Patrick. Thank you so much. As I said, we have recorded today, so we'll be putting up the recording on the ERA4TB video channel on the website, and you'll be able to we'll advertise that on our social media channels afterwards. So thank you everyone for attending today, and we will see you again soon.
Thank you to you, Kat, for organizing this wonderful webinar. Thank you.
Thank you.
Thank you.
Bye.
Bye everyone.